טוב, שלום לכולם. קודם כל, תודה שבאתם. נתחיל בעברית. נו, בשביל מה עשית את זה? אוקיי. זה התפקיד שלי, לעשות בלגן. מה? לא יודעת איך עושים את הדברים האלה. בסוף זה לא עובד.
למה לא עובד? עובד.
עובד? טוב. אז קודם כל, שלום לכולם ותודה שבאתם. והאמת, כמו שאומרים, זה ממש מחמם את הלב, כי נורא דאגתי בגלל צפת שיהיו פה מעט. אז באמת תודה. וברשותכם נעשה את כל המושב הזה באנגלית. So, Professor Lee Hood could understand as well. So thank you all for coming and joining us today for this lecture. It is my great honor and privilege to welcome here at TAU a very special guest, a unique and pioneer person and professional, which not always goes together. From all point of view, he received the U.S. National Medal of Science, a member of the USA National Academy of Science, National Academy of Engineering, and National Academy and the U.S. National Academy of Medicine. So just by looking on those three words, we see the combination of these three academic membership, his broad view of interest, and he's therefore the president and the founder of the Institute of Systems Biology that I guess everybody of you know. And it took, from my point of view, a lot of courage and strong belief to live then, in these days, a very well, safe position in Caltech and take your own road and established this Systems Biology Institute with, I would say, not few criticism and cynicism and all those schisms. He did it, and he was one of the leading then when he left in DNA, antibody diversity, etc., and decided to leave all behind and established what he believed was the right institute for that days. And today, it's proven very nicely. Combining biology, engineering, computer science, and so on. He did it despite, as I said, of all barriers, and he did it, and we talked about that today, the way that he decided to do it is as a private institute, not going through all the, should I say, via dolorosa of the academic world. I will share with you just, it's always nice, the stories behind. I share with you one small story where I met Lee about 10 years ago. He then was giving a lecture in Israel, and I thought, and I approached him because I thought he might understand me, because I felt like I'm kind of an outsider. Because at that days I was trying to preach for many years about metrics of biological knowledge and converging technology and other slogans, and it seems like nobody could understand that. And then I approached him, and he listened to me, and we walked on the beach, and he said, just wait a few years, everything will work, and you'll see. We did it already. It will come. And more than that, I was trying to say to him then, the systems biology then was up to here. And I said, we have to put in whatever we call as brain and mind. And he told me, I'm not sure. Today he really did it. So this is the systems biology, and I'm very, very happy that he is here with us. So, he gave yesterday a lecture at the President's Conference with a lot of panels, and you can see that there are still many that, how should I say, don't believe yet. But they will, because the paradigm, the change of the paradigm is there. So I'm very happy to invite Dr. Lee Hood to give his presentation. Thank you.
lots and lots of things. Well, thank you very much. Can you hear me okay? Okay. What I'd like to do is give you a personal view of how I came to systems medicine. And that... Oh.
What should I do?
I don't know. I'm a PC person.
Okay.
Okay, it's fine. It's fine. And that really started in my early years at Caltech in the 1970s. I was at that time a molecular immunologist, and over the next 10 years, I became increasingly convinced that we weren't going to solve fundamental problems in immunology one gene and one protein at a time. And of course, at that time, systems biology and the tools that we have today didn't really exist. But I did come to realize that biological complexity was enormously daunting. And I would say a big part of biological complexity is just the process of Darwinian evolution. It's a random and a chaotic process. It builds solutions on top of pre-existing solutions, and it makes solutions that are incredibly complicated, Rube Goldberg-like in design. And of course, there's also the idea of the fail-safe systems that have to be built into biological organisms, and they add an additional incredible layer of complexity as well. So with all of these in mind, I was wondering exactly how we were going to penetrate this whole area of complexity. And then again in the early 70s, I read a book that was really transformational in my thinking, and this was the Structure of Scientific Revolution by Thomas Kuhn. And he talked about paradigm changes in physics. And a paradigm change is an idea that transforms how we think about a discipline. And he pointed out that paradigm changes are incredibly hard to achieve because, in general, scientists are incredibly conservative. And we're conservative basically because of the way we're taught. We're taught to a certain extent that we know the truth when everything we learn is basically a hypothesis. So I had the good fortune to participate in 5 paradigm changes. In retrospect, they were my attack anyway on biological complexity. So in my early years at Caltech, I started bringing engineering to Caltech, and we ended up over 25 years developing 5 different instruments, including the automated DNA sequencer. And this was the forerunner of high-throughput biology in the generation of big data in biology. The automated sequencer and the development of it in the late 70s and early 80s got me invited to the first meeting that was ever held on the Human Genome Project in Santa Cruz. 12 experts were invited. We came to 2 interesting conclusions. One was that it was technically possible, although at that time in 1985, very difficult. And the second was we were split vehemently 6-6 on whether it was a good idea. And the 6 who were against it were fanatically against it. They felt it was big science, it was bad science, the Human Genome Project. Human genome had a lot of junk. Why would you want to sequence it? You'd never be able to get good people to work on it. And every one of the arguments could be met with the same kind of counters that we give today, but people hear what they want to hear. It's very difficult to get people to change their minds. And of course, what turned the whole project around, when I went out into the community, I would say in the beginning, 90% of the people were against the Genome Project for very much the same reasons. And of course, the National Academy report in '88 turned the whole thing around, and the Genome Project did start in '70. The third paradigm change I was involved with came about as a consequence of developing the automated sequencer, where it really required bringing together first-rate chemistry, good engineering and computer science, as well as molecular biology and enzymology. And that led to the idea that biology needed to incorporate the other disciplines into it so they could focus on developing the technologies that were essential to biology. And I proposed this to Caltech, and the biologists utterly opposed it vehemently, although the chemists and the engineers and the physicists thought it was a great idea. But Bill Gates made it possible to set up the first cross-disciplinary department at the University of Washington Medical School, and it really had a spectacular history in developing pioneering technologies in proteomics and cell sorting and computational aspects of the Human Genome Project. And I developed there the inkjet technology that Agilent has subsequently commercialized for large-scale DNA synthesis. The next endeavor was systems biology. I had been hired on to University of Washington with a promise after 4 years I could build systems biology on top of my cross-disciplinary department. The dean, serendipity plays a really big role in our careers, I'll tell you. The dean of the School of Medicine who made that promise died in an avalanche in the Himalayas. And the next dean that came along had a very different point of view about what he wanted to support. And it turned out for me that was really fortunate. Now, unfortunate for the dean, very fortunate for me, because it made me realize I could never do what I wanted to do in the university. There were a lot of bureaucratic hindrances which with a good leader could have been circumvented, but with a leader that had a different point of view made it impossible. So I set up the institute. It was the first Institute of Systems Biology. There was enormous skepticism at this time, 2000 or so, as to whether systems biology was real or was it just hype. And I think we've demonstrated that it was real. And of course, the final paradigm change is what I'm going to talk about today, systems medicine. That is the application of systems thinking to disease. And I'll show you how I think it really is transforming medicine. But before I do that, let me give you the lessons I learned from going through each of these 5 successive paradigm changes. Each of them really did fundamentally change not only how we thought about biology, but in most cases how we practice biology. In each case, there was incredible initial skepticism. And it was always this cry of, it's too complicated, we don't know if, let's put it off until tomorrow when we'll be able to deal with issues and things like this. And, you know, starting bold new projects is the way you develop new technologies and new lines of thinking. You don't do it by just sitting there saying, woe is me, we can't really get started. I think the third point that is really critical is this idea that each of those 5 paradigm changes required new organizational structure in order to be achieved. It is really hard to create new ideas, new paradigm changes in old organizational structures because the bureaucracies are created by the past, and generally they can just barely deal with the present, let alone think about dealing with the future. And I think the other thing that I'd really emphasize for people who want to do this is it really does require taking risks. And if you take big risks, it's always great if you're right. But anyway, so let me say, give you just 2 slides about the Institute to give a flavor of what we are. We have a staff of 230, 10 people. We're enormously cross-disciplinary, and we're about inventing strategies and technologies and analytic tools for system science. And we're focusing on human health on the one hand and on environmental sustainability on the other hand. We're really committed to transferring knowledge to society, and we do it in a lot of different ways, but we're really good at innovation. And in the last 13 years, we've participated in starting 17 companies in the Seattle area based on an algorithm that I'll talk about in just a few moments. With regard to strategic partnerships, they are all about enabling big science. And the classic example, one we did about 4 years ago now, was with the Grand Duchy of Luxembourg, where they gave us $100 million to develop the strategies and technologies of this thing I call P4 medicine. And in return, we established an institute for systems biomedicine at the relatively newly emerging University of Luxembourg, and it's been remarkably successful over the last 4 years. And just as a validation of quality, there is a Spanish institute that developed a metric for a series of metrics for evaluating papers about 6 or 7 years ago. And the most recent analysis they did last year, they evaluated almost 3,300 research institutes, and ISB's rank in terms of excellence, impact of its papers, was actually 4th in the world. And we had papers in biology, in medicine, in technology, in mathematics, and computation. So it shows you the power of this cross-disciplinary strategy. So let me give you a thumbnail sketch of how I view systems medicine and what are the essential features of it. Again, disease is incredibly complicated, and one of the ways that one can deal with complexity is, of course, to generate enormous amounts of data on the object that expresses that complexity. So one big essential feature of systems medicine is this idea in a 5 to 10 year period, every single patient will be surrounded by a virtual data cloud of billions of data points, and will have the wherewithal to reduce that data dimensionality to simple hypotheses about how to optimize wellness and how to minimize disease. And we'll talk more about what that's all about. These data are enormously heterogeneous. They range from molecular and cellular and systems biology data all the way up to social networks. And there are two big challenges. One, how do you integrate these types of data together so that you can create these predictive models? That's one of the grand challenges of systems biology today. And two, an equally big challenge, I would argue, is all large data have enormous signal-to-noise problems. And in biology, you can't circumvent the noise merely by machine learning, and I'm going to talk about that a little bit later on. A second point, one of the ways that we begin to reduce the data dimensionality is what I call the network of networks. And this is the idea that networks exist in biology at the DNA level, at the RNA level, at the protein level, at the metabolome level, at the level of cells, at the level of organs, all the way up to individuals and their networks. And these networks form an integrated whole. And when you get a disease perturbation caused either by genetics or by the environment, that alters the structure of the network and the type of information it can express. And this changed information is what manifests the pathophysiology of the disease, and being able to extract that changed information is really key to new approaches to diagnosis and to therapy. So let's ask ourselves, if we take a systems approach to deal with biological complexity, what are the aspects that let us deal with this complexity? And I would say there are what I call 5 pillars. So first is viewing biology and medicine as an informational science, and that's absolutely critical to being able to deal with the complexity and to being able to deal with the noise, and I'll talk more about that later. The second is, I believe you have to create an infrastructure and a culture that is going to enable you to attack in a cross-disciplinary manner this idea of biology necessarily driving technology, and that in turn driving new analytic tools. I'll say more about that in a moment. A third pillar is, one thing that's key to dealing with noise is being able to look at the dynamics of disease. That is only rarely possible in human beings for reasons that are obvious. Therefore, designing good experimental animal model systems, and we use mice for this, is absolutely essential because you can do the dynamics there and transfer the orthologies to an understanding of human disease. And I'll show you one example of this. We've pushed emerging technologies, and I'm not going to talk very much about that. I'll point out three I think are really going to be key for the future. But I want to talk about system strategies, new approaches to dealing with the complexity of diseases. And finally, I'll mention just very briefly the pioneering of analytic tools. I think as I go around the world, the biggest shortcoming in most places trying to do systems medicine is the bioinformatics. They don't really understand what you have to be able to do, and I'll just give you examples of what we've done. So what about biology as an informational science? So as you all know, there are two fundamental types of biological information. There's the digital information in your genome, and there is the environmental information that impinges on the genome from the outside. And it's the integration of these two types of information that create the phenotype, be it a normal phenotype or a disease phenotype. What connects the information with the phenotype are, again, two different dynamical structures. One are biological networks that capture and transmit and integrate and modulate and finally pass off information, in some cases, to dynamical molecular machines that actually execute many of the biological functions. And I would say the heart of systems biology today is very much in capturing the dynamics of these processes and then correlating it with the disease pathophysiology and disease mechanisms. And of course, the final point is that biological information is enormously hierarchical, starting with DNA and its modifications and epigenetics, to RNA and proteins and metabolites, down to networks and interactions and cells and organs and individuals and populations. And of course, the really key point is at each of these levels of information, the environment impinges upon and alters the fundamental digital signal for a particular system. Hence, if you're to understand a system, say, at the level of the cell, in theory, you'd like to capture all of the antecedent levels of information all the way back to the DNA in a global manner. And you'd like to be integrated, you'd like to be able to integrate these types of information in such a way that you explicate the environmental contributions as well as the genetic contributions. You'll never understand a system until you've delineated the contributions from both of these. The systems biology infrastructure was really at the heart of what I pushed in beginning to develop the Systems Biology Institute in Seattle. And the idea here is that leading edge biology always mandates that you develop new technologies that allow you to explore new dimensions of, in this case, patient data space. And once you've done that, the new data require the new analytic tools. And if you can succeed in putting together that holy triumvirate, you can really fundamentally change biology. And in order to be able to do this, you obviously have to create a cross-disciplinary environment with all the kinds of scientists we've listed at the right-hand side of the slide. And they have to be able to do two fundamental things. One, they have to learn to speak the languages of one another so they can communicate effectively. And two, they have to learn to work effectively together in teams. And we've taken 12 or 13 years at ISB to learn how to do this, and I think we're still in the learning process. If you can succeed in driving this circle, then not only does it lead to the discovery of new scientific concepts, but it leads to innovation as well. So you have new technologies, new algorithms, new kinds of concepts, and we've actually used these to spin off and create many of the companies that I talked about earlier. So the basic fundamental scientific process is also a fundamental process for innovation if it's done right. The holistic dynamical view of disease is really critical to, again, being able to deal with signal-to-noise problems, as I'll demonstrate for you. And I would say there are really two fundamental ideas in systems biology. One is the idea that if you're interested in the system, you acquire all the information that pre-exists, you build a model from that, it explains it, and then you create hypotheses to test your model. And you test those hypotheses through perturbations of the system, and in an iterative manner, you hopefully bring the data into juxtaposition with the experimental model and so forth. This is a process that both can be hypothesis-driven, and it certainly is a process that generates hypotheses, and its ultimate objective is to make models that are predictive, and in the case of patients, are actually actionable. Now, the second aspect of systems biology is how we view data. And I would say there are four fundamental elements here. One is you want to acquire data in as global a manner as possible because you don't know a priori what's involved in a disease process, so you need to look at everything. We can do that for genomics. We can't do it for most of the other omics. We need to be able to look both at the temporal and spatial dynamics of systems. That's absolutely critical. We need to be able to integrate the different types of data, as I've already explained to you. And finally, I want to make the point that in biology, there are really two types of noise. One is technical. That arises from the process of generating the data and analyzing it and so forth. And that can be handled in straightforward statistical and computational means. But the second is biological noise, and that can't be handled by machine learning or anything else. And I'll give you an example of what I mean by biological noise in this next example. So I'm going to give you a glimpse at a system we've studied for the last 8 years. It's prion-induced neurodegeneration in mice, and the basic idea is that you inject activated prion particles into the brains of animals, and they induce a disease that progresses over 22 weeks or so in a typical inbred strain. And what we did was to look at 10 different time points, to look at the brain transcriptomes at 10 different time points across that 22-week period, subtracting at each time point the normal brain transcriptome from the disease brain transcriptome so we could identify genes that are differentially expressed. And of course, to our horror, a third of the mouse genes were differentially expressed. So the signal-to-noise problem is absolutely enormous here. And we did a lot of things to deal with the technical noise, and that was the smaller part of all the noise that existed here. What we did then was to use deep and fundamental biological insights to construct a series of inbred strain, prion strain mice that allowed us to subtract away known features of biology that were irrelevant to neurodegeneration, because the brain transcriptome and its response is the sum of many different biologies obviously feeding into it. An example is we created a double knockout mouse for the prion gene, and that mouse, when injected with activated prion particles, never got the disease, but its brain transcriptome changed, and that was classic inflammatory response, and we could subtract that away. And we did that for 5 other things that I'm not going to go into. We ended up with about 300 differentially expressed genes we felt were coded into neurodegeneration, a more than 20-fold enrichment of signal to noise from the biology. We gathered a lot of data, the dynamical transcriptomes, dynamical histopathology. We looked at the spatial distributions of the infectious prions, and we actually mapped these things into protein interaction networks and gene regulatory networks as well. We did the clinical signs. And what we saw in looking at the dynamical histopathology was that there were 4 fundamental processes that were involved in this disease. One, something we call prion activation accumulation. A second, glial or astrocyte activation. It's basically a an inflammatory response. And then the third and fourth have to do with various aspects of neurodegeneration. So what we did was map the 300 differentially expressed genes into the protein networks that, the partial protein networks that express these various phenomena. And we came to 3 or 4 really interesting conclusions. First, these 4 major networks became disease-perturbed in a sequential manner. First, the most unique network, prion accumulation and replication, then glial activation, and then the 2 forms of neurodegeneration. That's important because if you're interested in early diagnostics or you're interested in therapy, the most proximal disease-perturbed network is a really fascinating one to begin your exploration. The second point was that when we took then the 4 networks and mapped all 300 of the genes in, only about 200 mapped into the 4 major networks. The other 100 defined 6 smaller networks and their dynamical behavior. These were networks that people had no idea were involved in the prion disease themselves. When you sum the dynamics of these 10 networks, you could explain virtually every aspect of the pathophysiology of the disease. And let me just say that we've done exactly the same thing with 4 different mouse models of glioblastoma. It's a brain tumor, and the general conclusions are exactly the same. I'm not going to show you all of those data. That disease-perturbed networks are seen well before clinical signs, so you can think about early diagnosis. Multiple networks become perturbed in a sequential manner. New networks are discovered by the global studies that are possible with transcriptome analyses. And these networks, for both diseases, explained much of the pathophysiology of the disease. And indeed, for neurodegeneration in mice, we've looked at several different types: Huntington's, frontal temporal dementia, as well as prion-induced neurodegeneration. We can actually beautifully distinguish them because each employs unique combinations of disease-perturbed networks. And the same was true of the various stages of the glioblastoma disease, too. And one intriguing thing that I think is going to be important for the future is cancer and neurodegeneration shared a fundamental network. And it's a network, actually, that's shared by many different diseases. And that's neuroinflammation in this particular case. What about emerging technologies and system strategies? So let me just talk about briefly a couple of technologies. At ISB, we actually have a spin-out from Intel that has developed a wonderful new technology that enables one to put together 2 different types of plastic material and to make pores of any size dimension you wish. And of course, they're focusing this ability in 3rd-generation DNA sequencing. That is, the sequencing using nanopores or nanochannels of single DNA molecules employing electronic signals. And of course, what is really exciting about these kinds of technologies is, number 1, you can hope to get very long sequencing reads, up to 100 kb. That means you can do de novo assembly of individual human genomes, and we'll, for the first time, be able to see really the structural variations that we lose with comparison against prototype sequences. Number 2, when the DNA goes through and you electronically record the individual different bases, you can encode any modification of those bases. So there are 16 different epigenetic marks on DNA that'll be read off, I think, in a straightforward way from this kind of a sequencer. And the approach that the company that is nested at ISB now is taking is one in which it will be as easy to read RNA molecules as it is to read DNA molecules. And of course, the final point is this process is infinitely parallelizable. So you could think about having a device that could record the human genome sequence in 15 minutes. and do it in very long reads and quite accurately. So that's what third-generation sequencing is about. Global mass spectrometry. There is a new instrument, a new approach in mass spectrometry called SWATH. I'm not going to go in and explain it. It's something that we're really pushing at the Institute. But what's exciting about it is it gives us, for the first time, hope that we can make proteomics at least somewhat as nearly global as its genomic counterparts are. And we can readily vision in samples being able to read out 6,000 or 7,000 proteins, identifying them and quantifying them, and or looking at chemical variants and isoforms and things like that. And a final point that I would make is that I think single-cell analysis is going to be one of the transformational technologies for biology in the next 10 years. And it's going to fundamentally change how we think about a lot of things. And the essence of what single-cell biology has done is it's shown us in tissues we think to be relatively homogeneous that there are discrete, quantized populations of cells that are interacting with one another in fascinating kinds of ways. Hence, if you take an organ and do a global analysis, you've lost the signal and you've increased the noise in this context anyway. So I think that's really going to be important in the future. So the system strategy I'll talk about are family genome sequencing, where we integrate genetics and genomics, a systems approach to blood diagnostics. I'll talk about one wellness assay that we're just beginning to work on, and then some examples of systems-driven large-scale Framingham-like approaches to clinical trials that use the data and strategies of systems medicine. And then we'll talk about ways we can stratify both disease and patients in interesting ways. So about 4 or 5 years ago, we decided to look at the sequence of a family of 4, and the parents were normal, the kids each had 2 different genetic diseases. And the question was, by sequencing the complete genomes of each of these individuals, could we reduce the dimensionality of the candidate genes that could possibly explain these syndromes? And it turned out that the answer was absolutely yes, but it did much more than that. We could reduce sequencing errors by 70% by simple Mendelian principles. We could identify instantly rare variants if 2 or more individuals had the rare variant. Couldn't be a sequencing error. And the most important point was, because we could map precisely recombinational sites, we could determine the haplotypes of the kids with respect to the parents. And that's really important because it allows us to reduce enormously the dimensionality of the chromosome search space within which disease genes must reside. We did the intergenerational mutation rate for the kids, 35 mutations per child. So that means there's no such thing as an identical twin, although whether the mutations are functional in any way is obviously open to consideration. And we were able to reduce the candidate genes for the 2 diseases from hundreds down to 4, and the assignment was a relatively simple and straightforward one. In this context, we developed an enormous pipeline for doing family genome sequencing and can very efficiently now take these data and use all of these Mendelian principles to do many of the things that I've listed here, and I'm not going to go into. We now have about 2,500 complete genome sequences from individuals in probably families that have 10 or 12 different diseases, and we focused very heavily on neurodegenerative diseases. And to give you an example, here's a neurodegenerative disease of childhood where we took 5 families, we did family genome sequencing, and we were able, from these 5 families, to identify 4 disease genes and then go to 18 sporadics and show 15 of the 18 had one of the disease genes. If you'd use these as random sporadic individuals, you would never have found a single one of these disease genes. So it shows you the power of family genome sequencing. So I'm going to argue we'll all have our genomes done in a 5 to 10 year period. And I think what's going to be a driver is we'll do it in the context of families, and we can use that to identify wellness and disease genes. And I think even more important, we've identified more than 300 actionable gene variants. That is, variants which, if you have them, we can tell you something you can do to improve your wellness. And an example of that was a friend of mine from Microsoft that started getting severe osteoporosis in his late 30s. He had an analysis done, had a defect in a calcium transporter, and he reversed this condition by taking 20 times the normal amount of calcium. So my argument is any rational person with these actionable variants would want to know whether they had one or more. And furthermore, because the number of actionable variants are increasing every year, once you have your genome done, they'll be investigated each year against the new actionable variants. So in a sense, your genome will be an investment in optimizing wellness over the rest of your life. And I think another driver is going to be this third generation sequencing. And my prediction is we'll have a $100 genome in a 5 to 8 year period. And that really is, I think, only constrained by funding for these third generation sequencing companies, which I can tell you is really hard to come by because some of the early third generation sequencing companies had, unfortunately, spectacular and expensive failures. So the VCs are somewhat skeptical. But I think we'll be able to get around that. Let's talk about systems diagnosis, making blood a window into health and disease. And a systems approach to disease involves many different components. Blood is really key. It bathes all the organs. The organs secrete molecules into the blood. And one of the classes of molecules organs secrete into the blood are organ-specific blood proteins. So these are proteins that have a specific molecular address, and I'm going to return to that in just a few moments. It's key to do longitudinal analysis because each patient is their own control for looking at change from normal wellness to transition and disease. It's really key to do multiparameter panel analyses because you want to be able to survey multiple potential networks that can become disease-perturbed. Quantification is important for obvious reasons. We've actually looked not only at proteins but at microRNAs, and both of them have worked very well for us. And there is an enormous amount of noise in blood diagnostics. In fact, the IOM had a report a year ago that said over the preceding 7 or 8 years, virtually all the papers that had published biomarkers, none of them have made it into the clinic virtually. There was maybe one exception. And the reason for that is they all employed the same algorithm. You looked at normal bloods, you looked at disease bloods, you really saw a lot of differences between the two. 99% of the differences are noise that are irrelevant to the disease process itself. So it's critical you have a way of dealing with signal to noise. And I'm going to show you how systems approaches deal with that noise extremely well. From a technical point of view and looking at blood proteins as biomarkers, targeted mass spectrometry has been really key, and ISB has been a pioneer in pushing this together with our collaborator Rudy Abersol. And two things. One, you can easily analyze in an hour 100 proteins in a sensitive manner with a dynamic range of 10 to the 5th or so. And number two, over the past 3 years or so, Rob Moritz at ISB has developed targeted proteomics assays for virtually all the human proteins. So when we have candidates, we can very easily convert them into blood assays. And in fact, Rob Moritz and Rudy and others have gotten coverage of the human genome, the mouse genome, and some other microbial genomes and so forth as well. So let me go back to the prion example in mice, and we'll use the brain-specific blood proteins as an example. We've identified close to 200 of these now. We have them both for human and for mouse. Those proteins map back into most of the major biological functions that we can define in the brain. So they not only give you pattern recognition of a change in state of the brain, wellness to disease, but they give you some insights into the biology of which systems are perturbed as well. And what we did was, in fact, to take 15 brain-specific blood proteins that mapped evenly into the 4 major disease-perturbed networks I talked about earlier, and then we followed the progression of the disease in the blood, and we were able to demonstrate 2 things. One, we could detect the first perturbations of the prion accumulation network 10 weeks before any clinical sign showed up, so we could do early diagnosis from the blood. And number 2, we could, from the blood, follow the successive disease perturbation of each of those networks, so we can follow very nicely disease progression. And I'm just showing you here a number of microRNAs from the blood that either were elevated or were decreased in this process. And I'm not going to go into it in detail, but the microRNAs are going to be incredibly powerful blood diagnostics as well. So let me give you the most complete example that we've done. This is with a company we started in 2009, Integrated Genetics, that employs these systems approaches to disease. And the idea here was the old classic question of lung nodules and what do you do about them. In the US, there are roughly 3 million cases annually, and the pulmonary oncologist is faced with what do you do with this lung nodule. And there are a series of different things you can do. On the one extreme, if you're convinced it's malignant, you can go in immediately with surgery. And on the other hand, if you're pretty convinced it's not malignant, you can do the watchful waiting for a couple of years. But in this intermediate range, where 600,000 cases a year reside, you can do PET scanning, you can do needle aspiration, you can do bronchoscopy. And none of those techniques are really completely reliable. So the idea that we were interested in is, could we develop a blood metric that distinguished a normal lung nodule from a neoplastic lung nodule? And let me tell you exactly how we went about doing that. We started with nearly 400 candidate proteins. We got them from comparing normal lung cells and cancer lung cells to identify differentially secreted molecules and differentially resident cell surface molecules. And let me say, in disease, a lot of cell surface molecules get cleaved and secreted into the blood. And then we went to the literature and captured molecules that were relevant from the literature. And we, of the 371, we were routinely able to detect very nicely roughly 190 of them in the blood from both normals and patients with lung cancer. So what we did then was to take plasma from 72 patients that had neoplastic lung nodules, as verified by histopathology, and 72 that had benign nodules, and we were very careful to match age, to match smoking experience, and to match nodule size, because those are 3 parameters that are used for decision-making process in lung cancer that we wanted to eliminate. We wanted to get an independent metric. So what we did then was to rank these 190 proteins with regard to how successfully they called the 144 different nodules. Then what we did, and this is really a key point, is we took the top 32 proteins and we divided them into 1 million panels of 10, and then we ranked each of those panels with regard to, again, how they called the 144 nodules that we'd studied. And then we asked the simple question, which are the proteins that consistently are found in the most effective panels. So these are, quote, cooperative proteins. And the cooperative proteins weren't at all the same as those that you rank ordered individually. So in a sense, this was a fundamental of a systems approach. We found 13 proteins that were highly cooperative, kind of made a break point in the panel, and we used those 13 in a validation study, independent benign and neoplastic nodules, and actually a new site as well. We have 52 cancer and 52 benign nodules that we analyzed, and actually the scores were even better in the validation study than they were in the discovery study. So Integrated Diagnostics will be commercializing this later this quarter. And there is enormous enthusiasm for a variety of reasons. It has a 92% negative predictive value, and that means we miss less than 10% of the malignant lung cancers. But what we can do, what we're really focusing on, is a rule-out test that covers more than 70% of the 600,000 patients that are in that dilemma range. You can do calculations on, if you can really rule those out, how much you'll save the healthcare system, because most of the surgical procedures are done on patients with benign nodules, since they constitute a much greater class than the neoplastic nodules. And of course, this gives the patient peace of mind. And to give you an idea, for the studies we did, the rule-out statistics that we have here are boxed. Again, blue, benign, red is cancer, and smoking, you can see on the y-axis. And of course, the size of the nodule is indicated by the diameter and so forth. So we can rule out very nicely, as I said, 70 or so % of the benign ones. And if you actually correct this for the real numbers of benign ones, you can see the incredible number of benigns that you can rule out. And again, I want to really emphasize the metric that we have here is independent for smoking, from the size of the tumors, and from the age of the patients and so forth. So it is truly an independent metric. The question you can ask then is, these 13 proteins, what do they mean biologically? And it was really elegant. 12 out of 13 map into 3 major disease-perturbed networks that are a fundamental part of the neoplastic process in lung nodules. So that means this assay has the ability to follow the progression of the disease. And in fact, what's really attractive is after 2 to 3 months, we can go back and do a second test to look at the very few malignant tumors that are in the rule-out, well, malignant and benign, that are in the rule-out category, and you can determine whether there's been any progression of the disease-perturbed lung cancer networks. A second area that we focused on is the diagnosis of brain disease in humans, and one we started with for a variety of reasons was post-traumatic stress disorder, because we had access to returning soldiers from Afghanistan, both normal and diseased, and I was interested to note that the disparity in record as to how many people had PTSD returning from Iraq and Afghanistan, namely 5 to 25%, reflected the fact that there were several neuropsychiatric assays that didn't agree with one another. So the idea that you could have an objective assay was attractive. So we created a panel of 10 that gave us a sensitivity and specificity of close to 95%. That's an ROC curve, one of the best that I've ever seen. And we're now in the process of doing the validation studies on a much larger population of soldiers, normal and diseased. And I think these really have important implications. First, we're also looking at individuals with traumatic brain injury. I think we'll be able to do the same kind of diagnosis there. and the neuropsychiatric diseases as well, schizophrenia, bipolar disease, autism, and so forth. And using these approaches, we're quite confident we'll be able to get markers for various neurodegenerative diseases, and we're pushing quite far along on Alzheimer's disease now. And I think these markers are going to permit us to do four things. One is early diagnosis. The second is following the progression of the disease. The third is following the response to therapy, and for neuropsychiatric diseases, that is really key. And then the fourth is it will allow us to stratify the disease into their different subtypes. So wellness assays is a big part of systems medicine, as you'll come to see in a few moments. And one of the most interesting wellness assays, and we have a whole list of them, and I'm only going to talk about the first one, are the organ-specific blood protein assays. And this is interesting because we've collaborated with Jim Heath now in making a microfluidic chip that has the ability to integrate in 50 different ELISA assays and analyze a fraction of a droplet of blood in 5 minutes. Again, highly sensitive range, good dynamic range and everything. And what we'd really like to be able to do in the future is to get 50 organ-specific proteins from each of your 50 major organs and then follow them for each of you longitudinally to see when there are transitions from wellness to disease at a very early stage so we can respond effectively. And of course, the idea of where you get 2,500 ELISA assays is a staggering problem. And that's why, again, together with Jim Heath and Integrated Diagnostics, we're looking at peptide protein capture agents rather than antibodies. And the idea here is Jim has actually constructed a library of 10 to the 15th D-amino acid 6-mers, and you can use the protein you'd like to make capture agents against to identify low binding monomeric 6-mers, and you can use a chemistry called click chemistry to join those together in the proper three-dimensional orientation so that you can make a dimer or even a trimer, and more recently we've made tetramers. With each mer you add on, you get about a log increase in sensitivity and specificity. Trimers give you sensitivity and specificity that mimics the best of the monoclonal antibodies. What is nice is these molecules are very small, 4,000 kD or so, so they're really ideal for thinking about in vivo imaging as well. And here's an example of a protein capture agent against human PSA. And when injected into mice, you can show it localizes beautifully to the prostate very quickly. And what we find exciting about this is it enables in vivo dynamics, both temporal and spatial, and we're thinking about things we can do with that. But I think these protein peptide capture agents have incredible features. One, they're infinitely stable. I think you can send them to Africa in an envelope. Two, they're sensitive. We can get trimers or tetramers that are as sensitive as you want them. And three, they're digital in the sense that if you know the monomers plus the coupling agents for each monomer, you can create large quantities of these molecules with copper catalysis in vitro in solution. We have recently learned how to be able to precisely direct these protein capture agents to particular epitopes, and I think that can avoid a lot of the cross-reactivity problems that plague most antibodies. And we think they can be adapted to large-scale production. So you can think about them in vitro diagnosis, in vivo diagnosis, or even possibly in the future as therapeutic reagents. And my gut feeling is in 10 to 15 years, if this goes as well as it is now, they will replace monoclonal antibodies. And we're actually spinning off this as an independent company to be able to focus the resources in developing it. Systems-driven large-scale Framingham-like clinical trials. These are really going to be key for medicine in the future. The ability to analyze thousands of patients, to do longitudinal studies where you look at molecular, cellular, clinical, and high-level phenotypic data, complete genome sequencing, and molecular analyses of the blood of the types that I've talked about earlier, and of course then be able to integrate it together and model it so you can identify the parameters of diagnosis or targets for therapy. We've done this with Inova, a community hospital in Fairfax, Virginia, where we've examined 750 families in a preterm birth study, done complete DNA sequence. We've done the blood analyses of proteins and microRNAs and methylation of the white blood cells and all these things. And we're just beginning the very large-scale correlative analysis of what is going to be able to predict those individuals susceptible to preterm birth. And we have some really exciting new data in that regard. It's still under analysis. And we're just starting a 7,000 patient atherosclerosis study with a company called Global Genomics, where the high-level phenotypic assay is high-resolution CT scanning that can give you the fraction of arterial walls that are covered with plaque, and correlating that then with genome sequencing and the blood analyses and so forth is something we're just getting started doing now at this point in time. What about disease stratification? We think there are three powerful ways for doing that. Taking patients' iPS cells, converting them in a test tube into the relevant tissue, say neurons, and then perturbing them with environmental signals, because each subgroup of a particular disease, such as Alzheimer's, employs different combinations of disease-perturbed networks. The environmental stimuli will give distinct readouts for each of the subgroups. Delineating the dynamic networks in the diseased tissues I've talked about, and the use of organ-specific blood proteins we've talked about as well. Another really key concept in systems medicine is this idea of stratification of patients into distinct groups that signal similar responses to environmental challenges. For example, there are probably 70 gene variants that make people susceptible to being able to deal with drugs effectively. Stratifying patients so they know which drugs they are not capable of handling, I think, is going to be key. And the same is true of heavy metal poisoning. There are enormous genetic differences in the responses there, and stratifying patients into those that respond and don't is going to be an important aspect of things. And the fifth thing we talked about is domain-driven transforming analytic tools. So Ilya Samulovich is developing tools for integrating all the different types of genomic data that come out of the Cancer Atlas project, and has done just a spectacular job in doing correlations that both allow disease stratification and in some cases potentially interesting clinical targets. Nathan Price is developing really powerful methods for integrating different types of networks, starting with the most accurate and the most fundamental network in some ways, because it's biochemically determined, namely metabolic networks, which can then correct the inadequacies of higher level networks and so forth. And finally, the Baliga lab at ISB has developed very powerful algorithms for predicting how a microbe will respond to environmental stimuli never seen before, and he's actually applied this all the way to cancer and done spectacular things with microRNA networks in terms of diagnostic predictions. So I'd argue systems medicine is really at a tipping point, and we've talked about many of the reasons. One, with the disease-perturbed networks, we can get at fundamental mechanisms, new insights into diagnosis and therapy. With family genome sequencing, we can actually identify disease genes and wellness genes. Transforming blood into a window for diagnostics. I think the algorithms we've developed will apply for any type of disease. stratification of diseases into their different subtypes is absolutely critical for a proper impedance match of the subgroups against their cognate appropriate drugs and so forth. And stratifying patients against environmental challenges is in a similar fashion really important. We've demonstrated by using organ-specific blood proteins, for example, in Alzheimer's, that the liver undergoes profound changes. And looking at multiple organs and how and why they change in diseases that we think are single organ diseases is going to be, I think, revolutionary. There are new approaches to drug target discovery using computational tools I won't go into, and increasingly we're beginning to focus on wellness. And this leads to a convergence then of the digital revolution, systems medicine, and patient-activated social networks. And the patient-activated social networks are really key because they're going to be the driving force for the acceptance of what I call P4 medicine. And what are the four Ps? Predictive. In 5 to 10 years, your genomes will give us enormous insights in optimizing wellness and minimizing disease. In 5 to 10 years, we'll have multiple organ-specific blood proteins where we can follow longitudinally transitions from wellness into disease. On the prevention side of things, we'll have new strategies for the identification of drug targets. And that's really key because drug companies are great at making drugs. They aren't so good at selecting the drug targets. And of course, wellness is going to be absolutely critical too. The personalized comes about from the fact that we differ by 6 million nucleotides from one another, hence we each have to be our own controls. And the participatory deals with the really important social aspects of this. How do we create the social networks that are going to be the driving force for accepting P4 medicine? And I think one of the problems we face that is absolutely fundamental is how can we have society agree to the fact that the virtual clouds of data for each patient have to be joined together in a way that can be mined by appropriate people to create the predictive medicine of the future that will transform healthcare for your kids and your grandkids. And how do we educate patients, physicians, and the healthcare community to P4 medicine, and how do we create a proper IT for healthcare? P4 medicine does differ from evidence-based medicine. It's proactive, it's focused on the individual, it's focused on wellness, it's all about mining, integrating, and modeling enormous amounts of data on individual patients, and it's all about stratification of patients into discrete populations, and not abstracting the characteristics of the patient and making bell-shaped curves and saying if you fall on the outer edges of the bell-shaped curve, you're sick. That just doesn't work. And of course, the patient-driven social networks. The two themes for P4 medicine are quantifying wellness and demystifying disease. And one of the really interesting social networks in the US is this network called the Quantified Self movement, started roughly 8 years ago by Wired magazine. And there are in many cities in the US now people who are using one or more of these 70 or 80 digital devices that measure your various parameters to give, through crowdsourcing, an optimization of wellness and how you do the wellness and everything. So there are two challenges for paradigm-changing P4 medicine. One is a technical challenge and the other is a societal challenge. And I think the societal challenge is far the greater challenge. And how do we see bringing P4 medicine to patients? One, through pilot projects with community and health centers like I've described earlier. And the other is through the creation of institutes for systems medicine, which will actually disseminate the vision. So I'll close with the 5 societal implications of P4 medicine. I think it will turn around the ever-escalating costs of healthcare to the point we can export it to the developing world and lead to a democratization of healthcare that was inconceivable 5 years ago. I think there's going to be a digitization of medicine that is going to make the data dirt cheap in exactly the same way the digitization of communications and information technology has made data really inexpensive, and that will be an important component of reducing the cost of healthcare. I think over the next 10 years, every sector of the healthcare community is going to have to rewrite their business plans in accordance with the dictates of P4 medicine. I think many of them aren't going to be able to do it, and there'll be enormous economic opportunity for new companies that can meet the demands of P4 opportunities. P4 medicine is going to create significant wealth, and I'll argue in a 10 to 15 year period, the wellness industry will far exceed the current healthcare industry, and that's an industry that's just getting started. There's enormous economic opportunity there. And it's going to transform the practice of healthcare, make healthcare better, cheaper, and enhance innovation. So lots of people have been involved, and I won't go through and discuss them, but thank you for your patience and attention.
So thank you very much. And now you see what I meant, and don't give up on your dreams. And I mean to the young people. And we'll have everybody who has to go because we were late, please, and somebody who wants to ask, we'll have like 1 or 2 questions. We see how I'm making. Any questions? So thank you very much, Lee, and I think people will approach you personally.
Okay.